Hello, everybody, and welcome to the broadcast of the This Week in Science podcast. We are here. Yeah, we, Blair, Justin, and myself, we are here to talk about science, all the science news from the week, and we're going to get right to it. What is in the podcast may not be all the things that we say here tonight, but it will be a very good representation for sure. So, are
There
we
won't be
ready for
anything
this?
new in the podcast. This is
Yeah, no, probably nothing
everything
added.
tonight will be in the podcast. There may be things tonight that don't show up in the podcast.
That's exactly
Omissions, not additions, is what we're dealing with.
it. Happy additions, no
Yeah.
omissions. Hopefully, we will not omit anything important. But 'tis time, so let us begin the show. Yes,
Yes.
we will do this in a whole countdown of a 1, 2. This is TWIS. This Week in Science, episode number 868, recorded on Wednesday, March 23rd, 2022. What should astronauts eat in space? I'm Dr. Kiki, and tonight on the show, we will fill your head with replication, entanglement, and mind control. But first,
Disclaimer. There are parts of your brain that are not you. It's your brain, and you are your brain. So yes, it's part of you. But the part of you that thinks that it's you is not the whole brain. That's just one piece in the brain puzzle. That's the cognitive, reasoning, active thinking, impulse controlling, list making, order of operation part of your brain that knows how to make breakfast. The rest of your brain is that of an ape. A great ape, mind you. A really fantastic ape, even. There are far more connections within the human mind than any other creature we know of. Far greater processing power is available too. We are walking, talking supercomputers with amazing powers of observation. But behind it all, just beneath the surface, always waiting to put us in our place, is the brain of an animal. A brain that is used to being the thing that's running the entire show, telling the rest of the biological machinery what to do, where to go, when to fight, when to flee, and when to fall in love. And right now, that part of your brain is telling you to fall in love with This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
and this good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again with science. We didn't bring anything else. Well, maybe we brought some conversation. We brought some good friends, but we have all the science. We're ready. We're scienced up. Are you ready? What did I bring? I brought stories about RNA evolution, entangled protons, and totipotent cells. Totipotent. What did you bring, Justin?
I've got just good news. COVID edition. For the second COVID edition, I think.
Oh,
I've
okay.
got remote controlled mice. PTSD goes clubbing. And the original people of San Francisco.
The original. Okay. I'm interested in that. I want to know. Blair, what is in the animal corner?
Oh, I have monkeys and ruffs, type of bird. And then I also have a quick story about lettuce. Space lettuce, specifically.
Space lettuce.
I'm really excited.
Oh, the space salad.
Yeah.
Anti-gravity, microgravity, it's how you toss it. Anyway, as we jump into the show here, I do want to remind you that if you are not yet subscribed to This Week in Science, you can find us all places that podcasts are found. We also broadcast weekly at 8 o'clock p.m. Pacific time on YouTube, Twitch, and Facebook. You can find us, look for This Week in Science on Twitch and Twitter and Instagram. We are TWIS Science, TWISCIENCE, and our website is twis.org, where you can find show notes, links to podcast episodes, and all sorts of information about the hosts and things generally to do with the show. But now, let's dive in to the world of RNA.
Ooh.
So
Tiny
DNA,
world.
the RNA world. The RNA world. It's like the idea, there's this idea that RNA could have been the evolutionary step prior to DNA, and that RNA may have had some mechanisms that similar to what we expect to happen through Darwinian mechanisms, natural selection, genetic mutation, all sorts of things occurring through the evolutionary processes that are present in our DNA that affect our phenotypes, the way that we look, and that those might have come from RNA, that DNA might not have been the thing to start it all, that RNA might have gotten everything started.
Would that mean that the first life form, and I'm doing air quotes there slightly because it's always debatable.
Right.
Would that mean that like viruses could be the first form of life on the planet?
Right. And that's the question. So what do we include in this idea of what is alive? Does it have to have DNA? Can it be an RNA-based organism? So researchers in Tokyo have for many years been looking into this question, and several years back they created RNA molecules that could reproduce. And so it's like, all right, they're not living, but now they're reproducing. They're copying themselves. And so that was like the first step, and it supported the idea that okay, if it's not just RNA there, this molecule in the wild, if it could have had this ability to copy itself, then that could have led to other steps as well. And so then the big question, the big question is, but how would it have evolved? Was there mutation involved? Could there have been molecular steps that could have led to the actual Darwinian kind of evolution that we think of in DNA, but in RNA? And so they created a situation with RNA replicase molecules, these molecules that replicate themselves, an enzyme that allows that replication to happen. And they incubated them for about 5 hours at a time at 37 degrees Celsius, which just happens to be human body temperature, 98.6 degrees Fahrenheit. Added nutrients, and they added a whole bunch of solution that mixed the proportions of different populations of RNA that were in this mix. And they repeated it over and over and over and over again. 1,200 hours of experiment. And the RNA molecules, through what they determined to be something of a predator-prey relationship, where one RNA molecule would mutate to a form that took advantage of the molecular capacities of the other RNA molecules, it would take over, and then it would shift back because it would overbalance the population, and then it had to shift back. And so they'd flux back and forth. These RNA kind of physiological chemical predators would boom and then bust, and very interesting. But the big take home is RNA mutated, diversified over time, and they wound up with 5 different RNA lineages that came from the single RNA precursor.
And again, this is why we only have one life form on this planet, right? Why we have a single basis for life? Because we've talked about it before, right? Like, whoa, what if there was a new microbe that somehow had been a new creation of life that came to be on this planet? And why don't we see that all the time if evolution can happen like this, or if life is this omnipresent in the universe? We talk about every niche of the microbes being filled. Now it turns out you even go down to free floating RNA and all of those niches are filled. So there's no where do you even start if you wanted to be a startup life form on planet Earth where you started from scratch and did the whole experiment.
You have to mutate and evolve some elbows. You gotta make elbow room.
Really sharp elbows.
Yeah, lots of sharp elbows in there.
And you're coming in billions of years after the thing started. It was like, we've seen that before. It's nothing. There's no new acts on the RNA stage.
Right. But the big thought here is that if this is a laboratory experiment, it's simulating, it was, put together in a particular way that led to this result. But the fact that it could happen, that the RNA molecules do have these propensities to respond to particular situations in a way that allows for mutation and diversification. This takes us back to this, this idea of, okay, let's put some more support in that early RNA life hypothesis, that maybe that's where it came from. Early molecules, chemical competition to RNA competition, DNA, replication, competition, the hunt for resources, and on, and increasing complexity until we have this beautiful world that we live in.
Yeah, and there's something, I don't know how related it is to this, but there is something in the
good.
Well, no, it's because it's post this stage of RNA just fiddling with itself. There giant viruses have been found to create novel segments of RNA within them, which is something we didn't think they could do for a long time, and then discovered in some of these really giant viruses that they were essentially just coming up with somewhat random RNA sequencing that they were creating as part of it. It's like, why would you do that? Well, part of it could be maybe that's like just happened into a lucky mutation at some point. But then it occurs to me, if they're in a community that are able to take maybe find an advantage in this, that's from a different virus, that might be the whole reason for it. Then it's like a sort of self-sustained feeding of innovation, whether it's the virus that's generating it or some other virus that's taking advantage of it. Anyway, I think I'm getting more and more in that virus first camp, and then
there's so many RNA viruses. It's great.
Yeah, with the pandemic stuff, we're like, oh gosh, maybe we're the invasive species on this planet after all.
That's right. I mean, everything is just framing, right? Who invaded what, where? I mean, what?
It's tough because viruses can't survive on their own. So the virus ancestors would have had to be more complex, right, to be able to support themselves. I think about how there's so many evolutionary mysteries that eventually come to light because of fossils. We're not getting fossils here. That's not an option. So we have to find another way to reverse engineer a timeline to try to figure out the most likely way that things progressed. But there's never going to be a smoking gun. Like people thought that whales were from an aquatic vertebrate that parallel evolution towards mammalian life. People thought that for so long, and then fossil record says it was practically a tiger. It was like a completely land-based thing that went back to the ocean. So and we only figured that out because of fossils. So this is something where it's the task at hand is so much harder to figure out what came first and all these things, because there's never going to be a clear until the time machine happens.
For the soft stuff, the only thing we can use is the genetic record, right? Mutational clocks and the genetic record to try and get ourselves back there. Yeah,
but be careful when you say never. We have that. We have the ability now to do ancient DNA and
We're
find
getting
out
there, but we're
flora
still
and
not
fauna
great.
and
It's
everything else in the
yes,
soil.
it's getting
Like
there.
we're getting, yeah.
It's getting better.
Some pretty crazy
I mean,
stuff.
seriously,
That's possible.
science
Okay,
never
I will
say
move on to my
never.
next
But
story.
let's move on to the
Just-
story. Yes.
Just good COVID news.
Okay.
If you have been looking for a silver lining in the devastating COVID-19 pandemic, this is it, maybe. Report in the British medical journal Open finds that in the UK, the COVID-19 pandemic reduced gastrointestinal infections. The number of reported gut infection outbreaks, and these are outbreaks, so these are clusters, right? Clusters of cases, more than halved in the first 6 months of the COVID-19 pandemic in the UK compared to the previous 5-year average. And lab confirmed cases, ones that actually got sent on to the lab, cases fell by a third. This is in the, again, open access British Medical Journal Open. So the data were split into different phases. They had the pre-outbreak was a phase 1, and then phase 2 was early outbreak. Phase 3 was lockdown. Then there was early lockdown, late pre-lockdown, early lockdown, late lockdown, lockdown easing. Like I think as they were doing this, it's and then further easing phase was phase 7. This is what we got, a 7-phase thing because of how this thing went down. But anyway, during the pre-outbreak, notified outbreaks were comparable to the past 5 years. First, pre-outbreak, that's pre-outbreak. Then the early outbreak. Now we're getting the news. We're being told, wash your hands. There's a virus thing out there. 22% lower than the previous 5 years. And the trend continued to go down and drop until we got down to 87% lower rates of gut infections by the late lockdown phase. And reports have remained substantially lower than historically observed numbers since. So breaking it down by cause, okay, starting with the pre-lockdown, they had reported viral outbreaks fell by 62%. Parasitic infection outbreaks fell by 94%. That's huge. So virus we're worried about, we start taking precautions, washing hands, wearing masks, social isolation. Not only did these other virus attacks fall by 62%, parasitic outbreaks, thing that we weren't even targeting, fell 94%. And bacterial infections fell by about half. So it's a correlation between lockdown and rates of severe tummy trouble. But as a correlative study, we're not actually talking about causality in any of this. But there's plenty of good guesses that can be made. Researchers point to improved hand hygiene as having a key role. People started washing their hands maybe for the first time ever.
Huh.
That was it. They also think that people may have suffered through some of the cases at home, were afraid to visit hospitals because hospitals
I was going to say there's that.
with COVID.
Yeah.
There was a reduced social contact, social distancing. There was more cleaning of environment. So there's a lot of things. My guess, which isn't specifically signaled out in this paper, restaurants.
Well, people were still getting delivery.
Well, I think
But
not
for
at
a-
the beginning. I was thinking
Yeah,
about
that's
this,
right.
and YouTube in the chat room 00 or 00 says home cooking. Yeah, and he's that
Yeah.
person is basically saying the same thing that you are, Justin. Which
Yes.
I know I waited months and months before I treated myself to takeout because it felt scary.
Right.
We weren't touching the mail for a while.
And then some
if you
other human.
recall. So somebody else touching our food.
Yeah.
felt
And somebody else who's
terrible.
out there in the world
Yes.
interacting to do this has touched the food.
Right. And early in lockdown, people weren't even going to work. Restaurants were shut. The people were not working in kitchens. They hadn't figured a whole bunch of stuff out yet. Yeah.
Mhm.
So people were just
Yeah.
cooking it. I think I've blocked it from my memory
Yeah.
at this point.
That first 6 months feel like a couple days now, right? Because it was
Yeah.
it was monotonous. Fada in the chat room is also bringing up another really good point. One of the symptoms of COVID is gut problems, is, diarrhea or vomiting or nausea. And so it could be that it was being reported as a COVID symptom and not as any of these other problems.
Yeah,
Interesting.
could be.
So that
But
is
yeah, if
very
you,
often when it comes to gut infections, there's drink fluids, replace the fluids that you're losing, try and minimize the discomfort. And there's not a lot until you figure out exactly what the problem is. There's not a lot that can be done.
Mm-mm.
So the treatment is fairly similar, regardless of if it's COVID or if it's, something else that's caused it. But that's interesting. There are probably many, many factors that went into it. But
and again, these are reductions of clusters of cases too. So that's also pretty amazing. So, but yeah, if you've ever wondered where your stomach issues have been originating from, and wondered if it had something to do with the food truck outside of the bar you went to after a night out at your favorite restaurant, you might be right. So it might have been something to do with one of those. Good news here is the things that we do to reduce infection work. And even though we were focused on one infectious threat, because we took those protections, we reduced others. Core prevention measures worked on a myriad of problems.
And I think what we're continuing to see now that people have started, people are going back to the doctor. They're not as afraid to go to the hospital, go to the doctor anymore for problems, and there is still a reduction. So, yeah, I would say that there are still a lot of things that we do, interventions that are still working. Keep it up, people. It's good for your gut. Good for you.
Good
Blair?
for you,
Yes.
everybody.
Yeah. Did you want to talk about something next?
Yes. Let us talk about space flight, shall we?
Oh, let us.
Let us please talk about spaceflight. This is a study out of UC Davis College of Engineering, and they were looking at what happens to bones in spaceflight. Bones are constantly balanced between growth and resorption, but in microgravity, it disrupts this balance, tipping bones towards resorption, and that's why astronauts lose bone mass.
Is it resorption, not reabsorption?
Resorption.
The scientists say resorption.
Okay. I feel like that's a made-up word.
It sounds made up, for sure. I agree with you. But so...
all words are made up at some point in time.
You know what?
No, some just stand on their own.
I think I grok that, actually. Anyway, looking at fixing the bone problems with lettuce, currently how loss of bone mass is dealt with in space, they have to inject PTH, parathyroid hormone, into astronauts regularly to prevent this from getting out of control. If you're thinking about long-term space flights, that's a problem for a couple reasons. One being, ideally, you wouldn't want to be doing a bunch of injections in space. If you have to, you have to, but it would be great to be able to do that orally. The other problem is that if you're talking long-term space flight, like to Mars, it will expire on the way.
Oh.
Right.
So how do you fix this problem? Lettuce. So UC Davis genetically modified lettuce to produce this drug that protects against bone density loss in microgravity. They developed a transgenic lettuce that expresses a function protein combining PTH with part of a human antibody protein. The fusion protein is designed to be stable in the bloodstream and to allow astronauts to potentially purify that drug from plant extracts. So their hope is that, for one, you get to grow lettuce in space, which in general, growing food in space actually is a morale booster on the space station.
I would imagine plants, greenery, and plants are also involved in cleaning the air and oxygen production and all sorts of stuff. Also, roughage. Yes. Good for your digestion.
So plants in space is good just for the brain and the air, but also for reducing the amount of time that you spend on the space toilet. All that to say, fresh food is good for astronauts. But on top of that, they think that they will be able to figure out how to engineer this lettuce not only to create this drug, but to have certain parts of the leaves that have this drug, certain parts of the leaves will be usable for food. And so it'll be kind of a double whammy. On top of that, they're hoping that they can figure out a way to synthesize this because of the combination of the PTH and the antibody protein. They think that they might be able to make this an orally available drug. So you might actually be able to take the lettuce and digest it orally so that you can get your PTH instead of via injections. As they have it currently, they definitely know that you should be able to extract from the lettuce to purify the drug to inject in their latest version of the lettuce. So they've made a huge breakthrough, and they're still going to keep going to try to figure out if they can use it for food and drugs, but also to be able to make this drug orally available. So
one of the issues with orally available drugs is that our gut likes to break things down. And so if you just have a molecule that's in the food, it somehow has to make it past that digestive process, all the high acidity in our stomach, the mixing and the digestion that goes on to be able to get into our bloodstream to get to the places where it needs to do the job it needs to do. Normally, something like that is going to get broken down into lots of precursor pieces, and our bodies will use it, but it won't necessarily be turned into the thing that you want it to be turned into.
Right. So step one is that this drug is stable in the bloodstream from the lettuce, but now you just have to get it into the bloodstream and make sure it is delivered appropriately from the gut. So right now what they're doing is they're taking the lettuce and then they have to put it in the food processor, and extract the drug. Yes, and then inject it. Which, even if that was all they got out of this, it would make long-term space flight possible in a way it's not currently because of the expiration date on drugs. So this fixes that problem alone. If you can then figure out how to turn it from an injection into an ingestion, and you can use it as food, and then you can apply this process to other drugs, that would be ideal. If you could have a floating pharmacy on the spaceship, that would be perfect.
We already do this. Tobacco plants here on Earth are used to produce all sorts of drugs, not just to be tobacco, right? So but taking this and making it work in space that's the key.
There's something of the if you're a hammer, every problem is a nail about this too. Is if you're in ag school, the solution to every problem is lettuce.
Lettuce and cows.
But that ag school approach to you're trying to preserve something that's going to last over months and months, and can you make a plant grow it? And then there you go. There's your sustainable solution.
Well, you want to put humans in space. You want to give them the tools that we've had forever.
Well, you want to make something last for a very long time and be powerful and one of the components of all molecules in the universe. Make sure you've got entanglement.
Oh,
yes.
Is that like my tangled hair?
Oh, mine too. It's just I got these split ends. I need to get my hair cut. No, not entanglement like your hair.
Okay.
No, this is the quantum physical process of entanglement, in which the probabilities of certain occurrences are linked between subatomic particles. Now, researchers from Mexico and Poland have published in the European Physical Journal C their theoretical model, which is now supposing that that physics happening inside the proton involves entanglement. So they did some very interesting experiments in which they shot photons at protons, which sounds like a pretty fun job to me. So if you're interested in physics, maybe someday you too will get to shoot photons at protons. Photons, electrons, when electrons which are negatively charged, impact with protons, which are positively charged, you have an exchange of charge and you have an exchange of energy, and photons are released. And so, really, what these researchers were able to do is use electrons and the movement of photons released during these molecular subatomic impacts to look at the trajectories of the photons to figure out what was going on inside the protons. Now, when I grew up, all the textbooks, they only talked about 3 subatomic particles: the proton, the neutron, and the electron. But oh, no. There are many more subatomic particles. Inside each of these subatomic particles are numbers of little tiny particles. Inside protons, you have gluons, quarks, and antiquarks. And these things interact in their own physical ways. And the researchers wanted to know what are the forces that determine how they're mixed inside of protons? How did the mixes happen? What happens in there? And so by using the photons, they were basically able to take these little pictures of the quarks, gluons, and antiquarks inside a small part of a proton. And by being able to see that little tiny part of the interior of a proton, they were then able to use probability to determine that the rest of the proton was probably made up in a similar way. And so what they've come to say is that the interior of protons is maximally entangled. Suffice it to say, this means that now if you see one part of the inside of a proton, this means that you can predict what the rest of the inside of the proton is like. If it's minimally entangled, you couldn't make any predictions. Maximal entanglement means that probability is linked very, very highly. So what you see in one part is also happening in another part. And so they're using little balls of light, little photons, to illuminate the interiors of protons to give us an idea of how they're made up, what kinds of particles are mixing together, how often. And this can give us even better ideas of the physics that is at work inside of everything. And it also links to things like the inside of black holes, which is another crazy concept because stuff going on inside of black holes is also entangled, and there's all this crazy stuff going on. There was another study this week that was just mind-blowing. 2 studies in which researchers were supporting Hawking's ideas. Well, actually not supporting, they were going against the black hole paradox, where we know that energy, or there's entropy, right? There's a loss of mass, and we know that stuff is leaving black holes, but we don't know how. And so now they have come up with an imprint based on a mark left in the gravitational field by matter collapsing into black holes that they call quantum hair. So now we've got entangled proton interiors and quantum hair, from the little scale to the big scale. It's all connected.
I require a rebrand. I do not like quantum hair.
No,
No.
Little
No.
But it's entangled. Come on.
Yeah. I get a wet comb.
I can't hear quark without thinking about the 10 forward. This in Star Trek.
Deep Space Nine?
Yeah.
When I was in high school, they were up quarks and down quarks.
Yes, there still are.
Okay. They're not the
And
same
charm
as quarks
quarks?
and anti-quarks?
Anti-quarks are anti. Yeah, those are different.
There's
There's
different.
Up, there's down,
There's
there's
an ad,
charm,
up,
there's
down.
strange.
Yeah.
There's like, what
This
is
is
it?
a lot
6
more
different kinds
than
of quarks.
See, you were saying you just had proton, neutron, electron. They threw in the up and down quarks for me, but that's all I got. This is all new. Oh boy.
Yeah. But anyway, there's a lot going on. I mean, you were talking, Justin, about never say never and our techniques and our abilities to see into the past are improving, but also our abilities to see inside of the components of matter are improving. The fact that we are able to utilize photons to be able to shine a light on protons is just
Yeah, and things
cool.
just are operating so differently at this scale. Just go Google how a gluon works and operates. It's the most insane, like counterintuitive thing.
It's not like Elmer's glue. No.
Well, no, it's this is, I think Feynman has this rubber band example, but the further apart you try to pull corks, the stronger their attraction to each other. It's like an elastic band that gets
stronger.
The further apart that things get. There's like, we fundamentally cannot picture what's going on at that level. Which means it's very likely it's not just three-dimensional interaction.
Right.
There's another dimensionality to it that is just impossible for ape brains to visualize.
I'm just thinking about how in high school, when I was first learning about the up quarks and the down quarks, that's also when, my teacher would say, okay, so light is both a particle and a wave. And I would go, why? And my teacher would say, it's because we don't know. It just is. It does that. And it seems like the closer we get to the, there's always another dimension deeper, right? And like, we're going to start to be able to build these very comprehensive answers. We know the way things are, but to your point, Justin, we don't know why, fundamentally why, for everything in the way that this stuff works. And so the deeper we look, the more we'll actually be able to answer the question of, but why?
Yeah, we can't answer things that we can sort of visualize. We don't have an answer for why gravity.
Right.
Like, why does gravity? Why does it even exist?
Why not? But thank you.
Shoot us into space.
Yeah. And that's something on a planetary scale that we understand very well. We can predict for the interactions of matter with gravity, not a problem. F equals ma, which is somewhere on this mug. Not a problem. That's it. We've had that one for a long time.
Yeah.
But we don't know why it works, really, or what's the mechanism of gravity is still
right. So, and Fada is saying there have been documentaries and other things. This quantum hair isn't new. The quantum hair is on the event horizon of the black hole, and while this particular concept itself may not be new, the researchers who have published these stories, though, is that they've demonstrated that with this paradox of having the quantum hairs, with having this connection between the interior and the exterior of the black hole, information is not necessarily destroyed, that researchers originally thought that it would require, and this is from one of the researchers who said that it would require a huge paradigm shift in physics, forcing the potential reformulation of either quantum mechanics or general relativity. What we found is that this isn't necessary. So there's no separation between the interior and exterior of the black hole. In the classical theory, the horizon acts as a perfect one-way membrane, which does not let anything out, and the exterior is therefore the same for all black holes of a given mass. And that's the no-hair theorem. However, in the quantum theory, with hair, the state of the matter that collapses and forms the black hole continues to affect the state of the exterior in a way that's compatible with present experimental bounds. Quantum hair.
So hashtag no bald black hole.
Justin, do you want to tell me about mind control? Laser beams?
Yeah. Why not?
I'd like to hear about that. That would be great.
You're supposed to say, I told you to tell me that.
I did. I hit the button and she said it. Neuroscientists. These are scientists from Neuro, land of Neuro. Neuroscientists for years have been messing about with optogenetics, which is a technique that controls the activity of neurons using light. One problem with doing this sort of research is the need to get the light to the cells, especially deep within the brain, where you want to do the activation. This requires some extensive invasive implants to be inserted and is generally considered uncomfortable to do if you were to scale it up from a animal research model to human. So scientists at the Wusai Neurosciences Institute at Stanford University have developed a method to excite targeted brain cells by making the skull and brain completely transparent. So that's going to be a nice benefit. Completely transparent to infrared lasers. Pew! Researcher,
not a supervillain. Got it.
Researcher Guzhang Hong explains that biological tissues, including the brain and even the skull, are essentially transparent to infrared light, which makes it possible to deliver the light much deeper into the brain. Since existing optogenetic tools don't respond to infrared light, Hong's team turned to a molecule that had actually been in biological evolution used to detect heat. By outfitting specific neurons in the mouse brain with a heat-sensitive molecule, TRPV1, the team found that it was possible to stimulate the modified cells by shining infrared light through the skull and scalp from up to a meter away. So this is like a good...
That's 3 feet.
Yeah, it's a good distance. That's a good distance.
Yeah.
And this is they were able to. So there's still some steps here that makes it sounds like it makes it a bit on the invasive side because you got to get this molecule to the right neuron. But then they also have this other aspect to it. So let me go on over here. So TPRV is a molecule heat sensor that allows us to feel heat-related pain as well as spicy stuff, burn of a chili pepper, that kind of thing. That's this molecule is involved in those sensors and in us, in humans. There's discovery of which was a Nobel Prize in 2021. Similar receptor gives rattlesnakes and other pit vipers their heat vision. In a recent study succeeded in giving mice the ability to see in the infrared spectrum by adding TRPV1 to their retinal cone cells, which isn't really part of this story, but I want to try that.
Yeah, gimme.
I know. I want to have heat
Turn them on.
I'm missing normal color vision. Give me that.
Yeah. Oh, yeah. You first. Yeah. You're like half blind. So new techniques also rely on this engineered transducer. This is another molecule that can be injected into targeted regions of the brain that absorb and then amplify the infrared light penetrating through the brain tissue. So it's called MINDS for macromolecular infrared nanotransducers for deep brain stimulation, MINDS. So there's another step that sounds a little bit invasive currently. Hong's team first demonstrated that their technique by adding the MINDS and the channels, the TRPV1 channels to neurons on one side of a mouse's motor cortex with the infrared laser, and the mice started walking in circles, driven by the one-sided stimulation of the motor cortex. In another experiment, researchers did the same sort of stuff, but they inserted the TRPV1 channels to the dopamine-expressing neurons in the brain's reward center. Okay, this gets really interesting, right? Now they've got this heat-sensing molecule on the dopamine-expressing neurons in the brain, and they set up a maze that had a beam of infrared light just in one section of the maze. Guess where the mice hung out the whole time they were in the maze? They spent the whole time under the infrared beams that were exciting their dopamine-expressing neurons.
Awesome.
Under the disco ball.
Yeah.
Mm-hmm.
So this is also
I like it.
you can picture the vast array of potential treatments just something like this could have by being able to locate these in specific parts of the brain that need additional excitement or engagement, and a non-invasive way, at least when you go to visit your doctor, to trigger them. One thing is you might have to, like, how do you avoid natural infrared light? Perhaps it just needs to be avoided to a degree, Like, I think going outside in the sun probably wouldn't necessarily trigger it, but having that little laser within a meter could. There's all sorts of great great ramifications for a dystopian future society where everyone is laser controlled in their brains, but we're not there yet. Hong and colleagues are also developing, they're like figuring out every aspect of this. This is amazing. Are also developing nanoscopic beads that convert focused beams of ultrasound into light, which can be injected directly into the bloodstream and should be able to go to targeted areas of the brain, which then can, and also it sounds like they might even have a way of moving them from one location to the brain to the other, so that they can change the targeting of their experiment in real time. So this is one of the things that they've developed.
I'm squinting my eyeballs and scrunching up my face because I'm thinking about how unlikely that would be. How are you going to move little photo beads through the brain without damaging a whole bunch of tissue?
Right. It's not, I guess, not necessarily that they would be moving the
but maybe they're moving in the
beads,
bloodstream.
But so it gets through the bloodstream and they can target where it's supposed to end up in the brain. That part's the thing. I don't know. They don't.
Fair enough.
But but it's not that I think they would move the beads around in the brain once they're in there, but that they can adjust where they're refocusing the light.
Just like put beads throughout the whole brain, right?
Picture a little
that's the disco ball. I mean, the disco ball.
Yeah, a little disco ball that can shut down some of the mirrors, right? Okay. That shuts down most of the mirrors, and then light comes in here, and then you can focus where in the brain it's going to go.
Yeah.
So they can do some very. One of the things that their first plan on using this for is then to be able to study the brain of animals while the animals are exhibiting a behavior. So the idea of this is real-time monitoring, not in the experiment where we're doing remote control, making the mouse go in a circle. That's just a proof of concept that you've got the interaction there. But this would be used for real-time monitoring of brains. So when you're doing the rat experiment, instead of having to, on some level, anthropomorphize or try to put yourself in the mind of the mouse. What was the mouse thinking? What does this mean that the mouse ate the cheese? You can actually register in real time what areas of the brain are being activated, what those processes, what cells at least are being triggered in their interactions. So really interesting.
Very cool. Minds control. It's really minds.
Yeah.
It's mind control, but because of their acronym and all that. I have robot hands.
You do? Since when?
They have been replaced. Yes. Researchers are working at Carnegie Mellon University to create a new system to teach robots how to use hands. And to do it, they are, guess what? Going to YouTube.
Oh.
Because lots of people show their hands and do different things using their hands in videos on YouTube. And they discovered that it's a lot more efficient to just come up with an algorithm that figures out what hands are doing and picks a bunch of movies and shows them to robots than actually having real people doing movements with hands to teach robots what to do. So it's much faster if you let the machine learning make the learning happen. Yes.
I don't know what's happening right now. This is not an interpretive dance. This is if you ever go.
This is what it looked like. It's really
Blair's doing interpretive dance to those of you on the podcast.
If you ever go to Disneyland and look at animatronics, they're on the whole they're very good, but their wrists are way too fluid.
Oh.
And so that's what I was doing. So those robots needed some time to learn how to use their wrists.
Articulate a human hand.
Yes. Yeah. So the robots, like normally the researchers say that prior work in this area relies either on gloves, motion markers, or calibrated multi-camera setup. Instead, our system works using a single uncalibrated camera since no calibration is needed. The user can be standing anywhere and still successfully teleoperate the robot. So they've published this in archive.org, and their YouTube trained robots are then able to be used by people and are pretty good at the actions, at the movements that people use. So now for telerobotics, for people in different locations wanting to manipulate objects through robots in other places over the internet, the robot isn't necessarily connected specifically to that person, but they've learned the general movements that hands make. And so that is the paradigm in which a user can come in and stand in front of a camera and try and make some hand movements and cause a robot on the other end of the cable to make similar movements and achieve a goal.
On the other end of the cable, I love it. But yeah, because once there's an interface, that interface can be, right next to you, connected to your hand through a cable, or it can be half a planet away or on Mars. Yeah, there's a little bit of a delay there, so that might be
a little bit of a delay, but still, this is the kind of thing that astronauts could use. It is the kind of thing that people in clean labs could use. You wouldn't even have to go into the lab to do some of
work from home.
You could work from home to do some of the dangerous research that you want to do.
But then isn't that also doesn't that
sensitive techniques?
All of those jobs that robots still couldn't take because it required humans to at least have nimble fingers to do things. Now those are going to go away too.
But they're not going to go away.
Yes, they will.
Really fast. I mean, let's just take. I just want you to take a look very quickly, and for the podcast audience, you're not going to get the full value of this video, but there's a video shared of the movements that the robots make, the robotic telekinetics. And in the video, on one side of the screen, you have a person making motions into the air, and on the other side, you have a robotic hand with fingers and a thumb moving to pick up and manipulate different objects. And although the user hasn't necessarily been trained how to use the robot, and the robot hasn't been trained in this one person's movements, they're very similar. But I will say that the robot does seem very clumsy. I'm going to say that our jobs are safe for the time being.
Yeah, that's tough. I also think it'd be really hard if you told me to do something so a robot did it, but I can't touch the individual items.
Right, you're not actually touching these things. You're just moving your hand in the air.
Some of the clumsiness might be from the robot, some of it might be from the interface. From the human. Yeah.
Because if you're just if you're making it up, miming the action, if you're not precise in your miming action.
Yep.
Well, the way that person just poured something, nobody pours like that. That was really
cheese
weird.
Cubes.
They poured it towards their chest, which I recognize is because that's the orientation of the arm. But that's so awkward. No wonder.
It does seem very awkward, but it's I don't know. Internet and robots, best friends forever.
It feels
robot, clean my room.
This could be taught or learned, sort of like you can't just tell AI at some point early. You couldn't just tell it. You show it one coffee mug. And then you could put a bunch of other cups up there and it wouldn't recognize them because it's not this coffee mug. It's only recognized this coffee mug as coffee mug. And this one, the second coffee mug, makes no sense to it. No idea what it is. And at some point you show it enough coffee mugs that it can kind of put it together. I feel like this thing just needs a learning curve and then it won't need the human interaction part. You can then just tell it,
I'm very excited. I mean, I have waited for this day when I can lie in my bed lazily and tell the robot to pick up the clothes off the floor.
Oof.
Robot, you
I
pick them, and
oof.
all I have to do is lie in bed and shoo my hand at them.
I'm just really scared to see the Boston Dynamics robots with hands. That's too much for me.
Yeah, let's not go there yet.
No, I'm not ready.
I'm not ready either. But you're ready for more science because this is This Week in Science. Thank you so much for joining us for this episode of Science News Discussion. We hope we are bringing you the fun of the science, the insightfulness, the curiosity. We hope that you're learning and that you go out and learn more. But in the process of that, we also hope that you tell a friend about TWIS today. Make sure to help spread the word.
Mm-hmm.
And as we come on back here, I have to get my little robot. It's not a cube. My rectangle, my robot rectangle to get ready here and play some music because
Is it that
it's
time?
It's that time. It is that time. It's time for Blair's Animal Corner.
With Blair. Loves all animals.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels.
What you got, Blair?
Oh, I have some monkey business for you, or rather some monkey play. This is a study from Spain, Brazil, and the UK.
Why are you
You just went all Silence of the Lambs on us.
This is a laugh. It's a silly laugh. I'm in a strange mood. Oh no. Anyway, okay, monkey business.
Get to it.
A team of international researchers from Spain, Brazil, and the UK have looked at play in howler monkeys. They looked specifically at two subspecies of howler monkeys, the Mexican howler, Alouatta palliata mexicana, and the golden-mantled howler, Alouatta palliata palliata. And they wanted to see how play varies with age and the availability of resources. They measured the amount of time adults play with other adults and with juvenile monkeys within their groups, all based on other things going on in the group, like what kind of food was available, what kind of food they were currently foraging, and how prevalent it was. And they found that howler monkey play involves individuals in very specific situations, and that play might actually take the place of something like grooming. It is a social behavior. It is something that is used to establish dynamics and potentially diffuse tension. So when howler monkeys play, they hang from their tails, they make facial expressions and signals, they shake their heads, looks very silly. But this research makes us think it's not so silly after all. It's not frivolous. It is, in fact, something that has a very particular use. This is particularly interesting because play is exhibited in a lot of adults in howler monkeys. And so it's not necessarily a way to learn social behavior or a way to learn techniques of, hanging from tails or balance or anything else, because they're fully grown adults that are doing this. And it is a very energy costly activity. Howler monkeys are herbivores. They really don't spend energy. They don't have to, because they're getting all of their energy from plants. They have to eat way more plants than if they eat meat. And so that also means that every calorie they spend and every moment they are not foraging, there should be a reason for that. So that leaf-based diet really limits their availability to do frivolous things. So looking at 7 different groups of howler monkeys in the rainforests of Mexico and Costa Rica, they found that the amount of adult play is actually linked to the number of potential playmates, increasing in line with the size of the group. So the more complex the group, the bigger the size, the more play happened. They also found that adults spent more time playing with other adults rather than juveniles. So it was not a mentorship program. And adult females actually spent more time engaged in play than adult males, which is not what you would expect. You would expect females to play less because they have the very energy costly experience of growing a baby. So basically, reproduction in females is so costly energetically that it can't be a frivolous behavior.
And they're just eating their vegetables.
Yes.
Yeah.
They also found that play amongst adults increases in line with time spent foraging on fruit. So at first, you might be thinking, okay, so they would actually just play more because when there's more fruit, there's more energy. But in fact, their research found that was not actually what they... So that basically the fruit-based diet, if it provided them with more energy compared to their typical diet, they should also have observed adults engaging in more play with all members, not just other adults. But the fact that the juveniles don't pose a threat or provide competition means that the reason the adults are only playing with each other, that's likely a diffusion game. So that
they don't get in fights.
Yeah, exactly. If there's a disagreement over fruit, a highly valuable resource, that they can diffuse that with play, just like you would diffuse something with grooming in another primate species. So they think that play could be a mechanism for solving conflicts. That would also make sense for the increase in female play, because there are more females than males, and there is more competition for resources, space, mates, all this kind of stuff between females than in males in a howler monkey group. Usually there's way less males. Yeah. So it sounds like, despite our own perception of what play is and what it means and what animals do it for, this is actually fulfilling an important function in these howler monkeys in a way that we haven't really seen in other animals. So we want to kind of associate what we see as silly behavior as a very specific thing, but it's us imposing that on there, right? In reality, this is just a form of communication, and it could serve a really important role in their society.
Well, it just makes me think of, our formal play by way of sports. So we have in our human, this is definitely, expanding this idea to humanity, but we have sports in which teams from different counties, different countries can play against each other and potentially diffuse tension. Not that the Olympics did that this year, but you have that possibility of people looking at things from a different perspective, and the sporting match being able to diffuse whatever resource tension is going on, so that you don't actually have to fight. Another thing this makes me think about is humor instead of play, where you make a joke in an uncomfortable situation to diffuse the tension. It's kind of similar to that.
I've never done that.
No, never.
Of course,
me neither. Not ever.
Yeah. So there you go. Play could mean a little more, and has evolutionary roots that are pretty important.
We should be looking at this more. Why is it? And why is it? I love the delineation between the adults and juveniles, like why that differentiation. And there's definitely social reasons. There's evolutionary reasons. These are social animals. There is a specific reason. And it's kind of neat that they're starting to dig into that for this species. And it, of course, may not be applicable across all species, but it is for this one. And
yeah, maybe that's why siblings fight over Monopoly, so they don't get in fisticuffs.
So you don't end up actually hurting your sibling. Yes.
Yeah, absolutely. So play, games, humor, this could kind of extrapolate to all of those things to let you know there's evolutionary reasons for those things.
Cool.
Yeah.
Give us some birds.
Yeah, so moving on from the very straightforward story of, hey, play's kind of important, to a pretty complicated one. So bear with me here. But I wanted to talk about rough mating rituals. RUFF. This is a kind of bird. They're a kind of sandpiper, actually. And this is out of Max Planck Institute for Biological Intelligence, looking at the super gene variants of ruffs. So you may or may not know that ruffs, they have these vivid examples of intraspecific diversity. So they have these males that have very elaborate plumage, and they have females that look very drab, as is often the way in birds. And they have these 3 different morphs. They have one that is extremely dominant, about 94% of the birds, and those are called independents, is what they call them. Then they have satellites, which are a small portion. And then they have about 1% of the species make up what they call fader morphs. So ruffs, Calidris pugnax, very good, are they have leks. So they have these huge mating arenas where males strut their stuff, they perform, and the bigger the lek, the more females come to watch. And they kind of observe, they take some notes, then they go, that one. And they'll go and they'll pick a male to mate with based on this large arena of displays. So the independents, the more dominant one, they have these huge, feathery, almost headdress looking situations that Kiki's pulling up right now. And so this is the dominant because this is the selected trait. This is the thing that the females pick that they find desirable for whatever reason. It could be the sexy sun hypothesis, which is why we have peacocks. It could be that it's a demonstration of virility or strong genes. There could be any number of things, right? But so they select for the bigger, more beautiful, more dramatic plumage on the head and neck. The satellites, they have lighter plumage. They are smaller, and they usually hang out near an independent. So they hang out near one of the big pretty boys, and then they can be like a consolation prize if somebody else beats the female to the independent that they wanted. The fader males look exactly like females. So you guessed it, they sneak around the edges of the mating arena unnoticed, and they can every once in a while win over a female by just being nice and sneaky.
So they're like the sneaky wingman.
Yes. And so what the reason for this study, there's a couple reasons. So one is, how do you have this drastic difference within a species where you have these dramatic plumage showing up in the males, and then you just have random males that just look like females? They found out about 4 million years ago that a super gene emerged through a genomic accident. A DNA fragment broke off of a chromosome and reinserted itself in the opposite direction, which made the fader morph. It made the morph of this bird that looked like a female. And so you would think that would die off really quickly. It was a genetic mistake. It was not very successful. And so you would think it would die off. It has not. 4 million years, this thing has stayed. So why is it not being selected out? Why is it remaining in the gene pool?
Because it's successful. It's able to sneak in there.
Right. But in a pure numbers game, 94% of these birds are the independent morph. 1% are the fader morph. 1%. How can you possibly maintain numbers over 4 million years with 1% of the population? And it's because they found out through this study that there actually is an impact on females from these different variants. So before, it was expected that it was just a sex-linked trait, but through this new study, they found that it was not sex-linked. It was not anywhere on the chromosomes relating to sex. So based on that, there should have been an impact on females. And they did find that the females had less mating success, less survivability of chicks. So it's actually a lower reproductive success of females with the fader genes. So now I will ask again, why didn't this die out? Not only is it only 1% of the population, but the females have a bad time reproducing. So
but they
how
do
can
reproduce.
You pass it along?
Probability? Is it just probability?
It's a combination of things. So it's the fact that it's just this small number that just maintains, but in additionally, it makes the fader morph. It engineers it to be a small number. So they actually found out through modeling that if too many of them were this morph, the males wouldn't get mates because all the females would go for the fancy boys. And the ones who were sneaking around wouldn't be enough to maintain the gene pool. But because it is self-selecting to be exclusive, those males actually have a very high success rate in relation to their population. And that is the reason that this can continue. Yes, so the genetic conflict between males and females is what contributes to the maintenance of the fader variant. The less common faders are, the better their stealth strategy works. So, and the fader and satellite gene variants are not viable on their own. They have to be paired with independents. So that's the other piece there, is that a fader cannot mate with a fader variant. So these independents are carrying these other variants just by occasionally dipping outside of that specific variant.
Which would also then mean that their effect on the population overall would be greater than that 1% difference, because now you have this growing population. So it's really interesting, because this sounds like also an effective strategy then, perhaps, of population control. Like, to take it directly to Neanderthal, which is, of course, always how I have to do these things. Neanderthals, we look at the Neanderthal genes in the current human genome and say, these are associated with fertility issues, where it's harder to have children, which, if you're Neanderthal, was a great benefit if you're a hunter-gatherer in a small population.
You didn't want to be pregnant all the time.
You don't want to be pregnant. You don't want to be carrying children everywhere. At some point, you got this family like, well, let's go hunting. It's like, well, who's going to watch all these dang kids? We can't, there's too many. We can't put anybody down. So it's actually a great sustainability and survival strategy for that to have happened in Neanderthals. And it might be something of a population control within these birds. I'm kind of then curious about their environment, and if there was, a time in which overpopulation was a thing that was affecting them in some way. And then if that's this variant, as it came along, stabilized the population.
Well, I don't know about population size, but it definitely would help with the gene pool remaining diverse, which is another really important piece, right? So, and the other thing they throw in there is that the independent variant, this 94%, this dominant, beautiful variant of male, they have kind of run away with themselves in competing with one another. They want larger, they want more aggressive, they want prettier. And so this is all about competing within each other. They have become, quote unquote, operation blind to the other variants that are operating in their
females.
The
the female choice females, and also the other males.
the kind of the smaller looking satellite males. Oh, I don't need to worry about him, right? And so this allows for it to continue. And so one of the reasons I wanted to bring this story is that I don't remember what we were talking about last week, but there was all sorts of conversation happening in the chat room about, well, doesn't evolution tidy things up? Isn't that the whole point of evolution, is to get closer and closer to perfection and to kind of narrow down what's best for a niche? No, evolution's really messy. It's actually about diversity. It's about trying all these different things and seeing what sticks. Some things stick, some don't. It goes out and in and out and in and out and in. And so in this case, diversity created a better system for these birds than having one particular type. And that in fact, some of the research say there is no such thing as the fittest, which if you think about the whole evolution thing, that's one of the main lines. This is survival of the fittest. Yeah. There is no fittest. There are a bunch of different tactics that can manipulate or take advantage of a niche successfully. And there's room for different types when there are different ways of being effective. If there's one that's way better than the other, it's going to push the other one out. But there is, in a lot of cases, room for multiple strategies.
Yeah, and so that's also important to remember when you're talking about fittest. It's really strategy that worked.
Yeah, and it's a gene that flipped upside down accidentally, and it actually created something helpful.
Yeah.
I love that they also figured that out. They're like, oh, it's this gene that did it.
Yeah.
That is the morph. This is the thing that causes this male morphed to look like more like the females. And
somebody put this Lego in upside down, but it still fits together. How funny.
Still works. This show still works. I hope this show still works for you. This is This Week in Science. We hope we're the upside down Lego in your podcast feed. I would love to be that. And if you love this show, please consider supporting us on Patreon. Head over to twis.org and click on the Patreon link and support us monthly at a level of your choosing, $10 and more a month, and we will thank you by name at the end of the show. We really appreciate your support and cannot do this without you. Let's come on back with Justin. You got some science, don't ya?
I do. Scientists report on a phase 3 clinical trial of the drug MDMA, aka ecstasy or Molly, when combined with psychotherapy for the treatment of post-traumatic stress disorder, PTSD. Preliminary data that this is of a phase 3 clinical trial. Therapy works, even in categorically hard to treat patients. And it apparently worked twice as well as psychotherapy alone. Beginning in the 1970s, some psychiatrists use MDMA to enhance psychotherapy, despite the fact that there was a lack of formal clinical trials or U.S. Food and Drug Administration approval. Back in the 70s, for those of you who don't remember, people would hear about a thing and then they'd just try it. There was no really good access to information on the internet. And so people would just try the thing that they heard of, which might explain why so many people of a certain age bracket have been trying the alternate treatments for COVID. Maybe there's just kids from the 70s who were like, yeah, we used to hear about a thing from the kid down the block. It's like, I heard from my
I don't know. Yeah, okay.
But MDMA, this is Mitchell, who is is at the University of California, San Francisco, one of the researchers involved in the study. Says MDMA is really interesting because it's an empathogen. It causes the release of oxytocin in the brain, which creates feelings of trust and closeness that can really help in a therapeutic setting. Which is sort of interesting because then I'm like wondering, does that mean they're at ease from the stress, and so then are allowed to function mentally through a psychotherapy session, or does that mean they just trust their therapist more? They trust that process more. It's kind of interesting. But MDMA, this says, can help reconsolidate or process fear memories in an area of the brain called the amygdala. Now, this is important because amygdala is part of the old part of the brain. This is part of, like in the disclaimer, I was talking about the animal ape brain part of you that's not really you because it's used to being in charge of the thing that you think of as you. But that part of the brain is the one that also sends the signals for fight or flight response. So the classic scenario here is if you're a zebra, being chased by a lion. It's this amygdala fight and flight response that kicks in. The adrenaline increases the heart rate, stops digestion. Off you go, running away from the lion, narrowly escaping. And if you're a zebra, then you pretty much just go back about your normal day. You're eating the savanna grasses, swatting flies with your tail. Normal zebra, rest of the day. It's a bit of a shortcut because there is evidence that shows that animals do experience long-term effects from stress, but we don't see the really debilitating things like PTSD in a zebra. We don't see that form of obvious stress not releasing again in a zebra to where it can't function. Humans, we've got this amazing prefrontal cortex, all these other areas of the brain as well that have developed beyond the animal brain when the amygdala was at the core of those sorts of responses. We can do all sorts of logical assessments and reasoning. We can go, oh, lion. Oh, wait, no lion here. Just relax. Everything's fine. And if we saw a lion once, we sometimes, though, still see lions, even when they aren't. Or we don't see them, but the part of the brain is reacting as if they were still there, which makes it harder to choose how we react. So we got this new logical brain that still gets told to be in fight or flight mode by the amygdala, and it doesn't have a direct communication back to the amygdala that everything's fine, that everything's under control. So it's sort of like getting spam calls, scam likely calls, spam emails. There's almost no way to turn off the phone from ringing from the amygdala. And so it can be a really bad actor in continually sending stress response alarms when a logical brain doesn't need that. And worst thoughts and memories, parts of the modern brain. So we're thinking about a thing or picturing a thing, and PTSD, remembering a battlefield scenario or something of this nature, whatever the trauma is, and the amygdala can react to it as if it's still happening. So all this is a big tangled mess of misinterpreted primate brain to advanced brain happening. And so we're constantly, some folks with PTSD are constantly running from the lion.
Mm-hmm.
There are selective serotonin reuptake inhibitors. That's the first line of therapeutics for the disorder. They're effective in about half the patients, according to Mitchell. Many people with PTSD either fail to respond to that or just quit going to their psychotherapy sessions. So the team enrolled 90 people with severe PTSD for the first phase 3 randomized double-blind placebo-controlled study of this MDMA-assisted therapy. Participants attended an 8-hour therapy session after the dose. This process was repeated twice, a month apart each time, in addition to weekly therapy. 2 months after the final session, about 2/3 of the people who received this MDA-assisted therapy no longer met the diagnostic criteria for PTSD, which is an incredible number for a 2-month period. This is kind of even the drug with psychotherapy, of the drugs that are on the market, we're talking about years down the road to be able to have an effective rate of about 1/3 of the patients feeling a relief from the symptoms. This is 2 months of treatment and 60-something%.
It's incredible. This kind of stuff. I mean, you were talking about all the stuff happening in the brain and the blocked emotional cycle where the emotions from the original event get stuck, and that you're not able to move past the fear, the anger, whatever it is, and so that's the trigger all the time there. But yeah, the MDMA, it goes in, and thank goodness we have these plastic neurons and the synapses to remake connections. And so you go in and you you dig up these old traumas in a safe place, in a mental state where you now you feel safe. You've got the oxytocin, you've got the MDMA in your system. It's releasing serotonin. You've got dopamine release. You've got a lot of stuff going on to allow for real. And this isn't just, it's not a surface change. You're changing neural pathways. You're changing the memory in the brain and turning it from a triggering memory to something that you can handle. Yeah, and that's what MDMA allows.
It's also, it's very difficult because apparently, from what I understand, you know better than I, which I always feel confident talking about the brain on this show, because when I get it wrong, I know I got a brain.
No, you got it. It's good.
Like, that the amygdala, that emotional response that can trigger all this hormone releases, trigger the body to react in different ways. It's allowed to communicate to that modern evolution prefrontal cortex brain that has all that logic. It's able to send it all sorts of emotional signaling and tell it like what's going on. But that logical part of our brain has to use some weird roundabout back channels. We don't have like the direct communication back to try to regulate, because the amygdala is supposed to be a regulator. It's not supposed to listen to the rest of the brain. It's designed to like take that ape and make it run from a lion, or the zebra.
Save your life. It's supposed to save your life.
Override every thought that might be going.
Justin, I have to push back on your zebra analogy a little bit, because
please do. And I tried to put a little disclaimer, like, it's not exactly the story, but yeah.
No, I just the idea to characterize PTSD and stuff like that as a kind of frontal lobe, more human issue. I think I understand the need for specific drugs for humans with PTSD, but I also think that there could be applications in animals, because a zebra can be traumatized and experience PTSD, not from a lion, but from something they're not accustomed to dealing with. And so anybody who's ever dealt with abused animals that are afraid or overgroom or exhibit stress behaviors know that animals can have obsessive behaviors and stress behaviors related to trauma. So that's definitely something that is not unique to humans. And if it is something that is specifically affecting our brain, this is something that could actually be applied to animals as well.
That's a neat concept. Yeah, I love that, Blair. Thanks.
And there is also the less specific to the lion, but there's other evidence of when you have hares. It was one example I had seen. When there's certain animals, when a population is exposed to frequent predation, that they tend to produce less offspring. They tend to eat less. They tend to venture out less. So there are larger stress issues to having predators. If you're a prey species, that's part of your gig is going to be a little bit stress prone.
Stress. Yes.
Yeah. Right.
For sure.
But you can see a zebra chased by a lion, get away from it, go back to the herd, and then just go back to grazing. Like it's like no human would just be like, did you guys everybody see that? I almost got eaten by a lion. And then
but I can say the same to you about hearing a car alarm. You hear a car alarm, you go, huh? Oh, it's nothing.
Yep.
It's so we're accustomed to our own.
Me.
We're accustomed to our own stressors, like a shout, like a cry for help, right?
Whatever
so we're accustomed
usual.
to our own stressors that to other organisms would be stressful and scary and a reason for alarm, but to us is not, right?
And
I'm glad we don't have to deal with lions.
And very often PTSD is from stressors that are not the usual. It's not the stuff that people should be used to. So, yeah. What's your next story, Justin?
This is a fun one. Prior to European contact, the territory is now known as California. Actually hosted one of the most populous and diverse Native American communities on the continent. At contact, more than 15,000 Native Americans from 5 distinct language groups were residing in the Bay Area. Studies of the Bay Area's dense archaeological history reveal that intensive sedentary or semi-sedentary habitation extends back more than 5,000 years. And the region's archaeological record actually gets back to around 11,000 years. So there's been a long, long time of habitation there. There was an introduction of 5 Spanish missions that happened somewhere between the 17 and 1800s. Mexico claimed the territory in 1822. 1850, California became a state of the United States. And 1 year later, the governor of California promised a war of extermination will continue to be waged between the races until the Indian race should become extinct. Which is just to preface how San Francisco came to be. A genomic study of native peoples in the San Francisco Bay Area finds that 8 present-day members of the Muwekma Ohlone tribe share ancestry with 12 ancient individuals who lived in the region over a span of 2,000 years. Previous studies of artifacts and language patterns suggested that the Ohlone were relative newcomers to the region. So by relative newcomer, it would be about 500 years ago that they arrived. It wasn't what the tribe believed. It wasn't what their stories told. And for people who lost the homeland, had gone through forced conversion, genocide attempt, were forced into re-education centers and forced family separations that went on as recently as 1978, to then be told that their ancestral claim to the land wasn't what they thought it was in the first place. So it probably just seemed like another round of cultural extermination coming from anthropology. So it is somewhat amazing that the surviving members of the tribes were willing to even work with researchers, given the history. But they did. It was part of a community engaged research project. So they did some genomic research, found continuity, ancient population and the current one. They analyzed a large number of ancestral remains for DNA preservation and focused on the ones that had the best preservation for this study, according to Rupan Mali, a professor of anthropology, University of Illinois, Urbana-Champaign. We also worked with the Ohlone to sample saliva from present-day community members so we could compare the DNA of both groups. The team also took DNA from individuals who lived around California, other parts of North America, to look for genomic similarities, differences between the groups. The ancestral individuals belonged to 2 villages near San Francisco Bay that were excavated ahead of a large infrastructure construction development thing. So they're going to do this infrastructure construction for, I don't know if it was the city or the state or a private entity. Native American remains were found. And so at the request of the Muwekma Ohlone Tribal Council, Far Western Anthropological Research Group excavated the sites to get those things out of there ahead of this. And the Muwekma tribal members participated in all aspects of the archaeological fieldwork. They were the primary excavators of all the burials. Quoting Brian Bird, who is an archaeologist at the Far Western Anthropological Research Group in Davis, California, this was a rare collaborative, community-engaged research project with tribal members and archaeologists working side by side for more than a year of fieldwork, resulting in tremendous repatriation of knowledge to the descendant community, which is a very important thing because that's not how archaeologists used to operate. You would almost ignore, not just almost, absolutely ignore. I mean, this used to be like Indiana Jones. Hey, let's go to this place where there's this indigenous people who have this culture, and let's steal artifacts from their ancestors and bring them to a museum somewhere far away.
And ignore the people it came from. Yes.
And ignore
in a museum.
The tribal council also requested and approved a study design for the genomics work, and members of the study team regularly met with the council and enrolled tribal members to review the work that they were doing. So, I mean, that's that community engaged version of archaeology is the framework that is now part of the ethics of archaeology. Didn't used to be, but that is very much now the new model that's being used as much as it can be. So, yes, this was
cool.
Not, if I don't think it was the Miwok tribe, but it's the Honi. So it's also there. A caveat to this.
Yeah, the Miwoks were, when we were kids, the Miwoks were basically given all of California because it was easier to teach that way, but it's not the case at all. There's the Ohlones, there's the Ohlone Ramatush, there's
and like, the Ohlone in this region also is also an umbrella of again 5 distinct languages of peoples. Right. There was and these people who are related to the ancestors, they're saying they're related to the group of peoples that were there too. So it's not exactly tribe specific as far as the excavation site and everything. Again, a lot of this too has to do with the archaeological record, anthropological record, having been erased by genocide, cultural extermination, and all this over the last hundreds of years, because we could have a record of all this. We could know what tribe that is. We could know things about the language. We could know things about the origins of the people. We could know all sorts of cultural aspects of these people, except that they were basically, to a large extent, swept off of the earth in order to put San Francisco there.
Yeah, our missions. It was our mission.
It was past the missions. I mean, 1978 is when they got rid of the family separation laws. The ability to separate children
to me
from their parents to re-educate them. That's 1978. So let's not pretend this was ancient history.
Yeah.
Let's not pretend this is all the way back to missions even. This is very recent history.
Yeah. Erasing history is very recent history. Yeah. Well, this is This Week in Science. I have a couple more stories, and then we are out. Totipotent cells.
Totipotent.
Totipotent.
So I heard of the pluripotent.
Yeah, pluripotent.
Yeah. But what's the totipotent?
Let me tell you about the totipotent. So pluripotent cells are cells that have the ability to turn into just about every cell type in the body, but they have limitations. And because of these limitations, there are little genetic factors that have been involved that have held them back from really being the wealth of like the fountain of help that we have needed for regenerative medicine. So we've been looking at embryonic stem cells. It's like, whoop, we found these cells in the early embryo, and those can go to be those things, and those can go to the other things. But we don't want to use embryonic stem cells for everything for various reasons. And so being able to induce pluripotency is just this wonderful thing that we're able to do now. We can take a skin cell and turn it backwards and then use it to create a brain cell. Like stuff like that is amazing. Oh, we're going to change your form. But the problem is, like I said, there are these little molecular factors, genetic factors that occur very early in development of organisms. And to really get cells that can be anything in the body, you need cells that are from the 8-cell blastocyst stage of the early embryo. 8 cells. This is like early. Those 8 cells, after the first couple of divisions, they can go on to be anything. They're free. They are totipotent. They're totally potent. They can do all the things. But, we can't go around taking the embryos and just stopping them at that blastocyst stage. It is much better for a variety of reasons to be able to take that skin cell of yours and turn back the clock to that very beginning state. And so researchers from BGI Research, Chinese Academy of Sciences, have published in Nature their discovery of a transgene-free, rapid, and controllable method taking pluripotent stem cells, so like the skin cell, wound backwards through time to the pluripotent stem cell stage, and then wound backwards even further into totipotent blastocyst-like embryo-like cells.
So I looked it up because I wanted a reference point.
Yeah.
So in humans, at 3 days after conception, you're already at 16 cells.
Yep.
At 2 days, you're between 2 and 4 cells. So sometime between day 2 and day 3 is when you get your 8 cells. So that's quick.
It's
That's
very
very
quick. It's
Yeah.
right after fertilization. It's
Yeah.
it takes the longest time to get into that first division. First you have the fusing, and then you have the division. But then it's that second division. And then after that, things start happening faster and faster and faster and faster. But yes, these cells can differentiate into placental tissue. So could we do placental transplants? These cells can potentially differentiate into more mature organs. Maybe these cells would not have the tumor-causing potential that some of the pluripotent cells have had that we've come across. Because if you try and take a skin stem cell and then put it into the brain as a brain stem cell, maybe it won't quite work exactly as that brain cell should. But this totipotent cell,
Do what it's supposed to.
Yeah, it would do what it's supposed to. Exactly.
Nice.
Yeah. So hopefully this new process that they have developed, they've used single cell sequencing technology with what they say through large scale single cell multi-omics profiling. There's a lot of jargony words. But really they're able to look at single cells, determine their state and their tissue type, and be able to identify these cells that they can then use. Anyway, innovative technology. Again, technology moving us forward, but also potentially allowing us to move forward in regeneration, creating organs to solve the organ crisis.
With great efficacy, too. I love the idea. It's like, well, instead of having that conversation with the doctor, it's like, well, we got this treatment we can do that's going to help grow your liver there, Mr. Jackson. Oh, that's great. Let's do it. There's a chance that you might grow an ear instead. Like, you don't want to hear that.
I don't want an ear. I just want that liver.
No, I want whatever thing that I need to have replaced. I want that and not some chance at completely different.
Yeah, and then the additional bonus to this is that for researchers who do want to work on embryonic tissues, this potentially gives us a way to take that skin cell, turn back the clock to this totipotent early division state, and be able to do research without having to rely on actually procuring embryos. So there are lots of research and eventual good therapeutic outcomes from this. And then finally, because this was World Water Day week. I don't know.
I think that was yesterday or the day before yesterday. But yes, it was this week. World Water Day.
I don't know. A lot of things have had me thinking about climate change and the water on the planet and all the things going on. Researchers have just published in Science Advances their study looking at old expulsions of carbon dioxide into the atmosphere and what happened. So they compared a period called the Paleocene-Eocene thermal maximum, the PETM. It's about 56 million years ago, in which volcanic activity spurted a whole bunch of carbon dioxide into the atmosphere, caused extreme global warming, massive climate change. It was really, really bad for a lot of life on the planet. Took tens of thousands of years for the planet and life on the planet to kind of come back, for things to come back from all of that warming, they discovered that just prior to the PETM was a little event that had about the same amount of carbon emissions as what we've emitted so far from fossil fuel burning and other activities that we do. They compared what happened and how long it took for the planet to recuperate, for life on the planet to recuperate. And says one of the co-authors, James Zakos from UC Santa Cruz, there was a short-lived burp of carbon equivalent to what we've already released from anthropogenic emissions. If we turned off emissions today, that carbon would eventually get mixed into the deep sea and its signal would disappear because the deep sea reservoir is so huge.
And would be gone for like 50 years.
Yeah, it would be gone for a while. It would get mixed in. It would take hundreds of years. It would take a very long time for that deep mixing to occur and, for that extra burp of carbon dioxide to actually get evened out. So at a human time scale, it's still a long time and we don't really want to be dealing with it, but we have to. But globally, Earth geologic time scale, hundreds of years is very manageable for a lot of life on the planet. The climate system can recover, things can come back. The new findings, they based on an analysis of marine sediments in shallow waters on the Atlantic coast, part of the Atlantic coastal plain. At the time of the PETM, sea levels were a lot higher. And so they got these sediment cores, and the marine sediments that they used, they were able to get carbon isotope compositions to let them know what kind of carbon dioxide was being absorbed by the ocean. So like at the time that these sediments were being laid down, they were deep ocean. So it was giving them an idea of what kind of absorption was happening deeper beneath the sea, similar to what would be happening in the deep sea here during our time now. Foraminifera, little tiny sea creatures that have these carbonaceous shells, were in the sea sediments and were the records that they were able to look at. And the researcher Babala, Tali Babala, also at UC Santa Cruz, says previously thousands of foraminifera shells were needed for boron isotope measurement. Now we're able to analyze a single shell that's only the size of a grain of sand. Yeah.
That's great. I think the thing that you've said so far that's sticking out to me, though, is if we stopped today. Because I think, you think about the all of the carbon that we've released since the Industrial Revolution, it seems like we match that every 5 or 10 years because we exponentially grew to the point where we were the amount since the Industrial Revolution, that entire mass, we're just constantly doubling it, right? So the problem is that we need to stop. We still need to stop. Like, this is good news. It means that we are not past the point of no return, as so many people like to tell us all the time. But it's still,
don't listen to the naysayers.
We still have to cut off the carbon.
Yeah. So that number is absolutely correct. It's by decade, right? So in the 1990s, we used as much carbon in that decade as all the way back to the Industrial Revolution. So that decade doubled. In the 2000s, we did that, including 1990, all the way back again. So there has been this decade by decade doubling effect. So we're accelerating towards the
but still we're in the range of, so in this particular study, the short-lived precursor pulse, which was managed by the buffering capacity of the oceans. And that's something that Blair's talked about. We've talked about on the show a lot. Ocean acidification happens because the ocean is able to take in carbon dioxide, and there is a change in the hydrogen ion concentration as a result of that. This is simple chemistry. but the buffering capacity of our ocean is only so big. And so what happened in this historical case that this study is telling the picture of is that short burp was managed. The ocean buffered it. It took a long time. It didn't happen all at once. There was still a lot of carbon dioxide and warming in the atmosphere for a long time, but eventually the ocean evened everything out and the cycles reemerged. But when the volcanoes got involved and there was massive amount of carbon dioxide that outstripped the ability of the buffering system to manage it, that's where new processes became involved to bring everything back to normal. And then they started seeing processes like the weathering of silicate rocks, carbon dioxide and the silicates working together and buffering using more physical chemistry. And that took tens of thousands of years. Life still survived. There was still life. So it's not the end of everything, but our humanity and what we're doing, yeah, I'd much rather deal with a few hundred years than the tens of thousands that we might not make it through.
Yeah, it's back to that thing we keep saying, that the Earth is going to be fine.
The Earth is going to be fine. Life will be fine. It's just which species will be fine.
Yeah, that's really the question.
What do we really care about? If we're taking a cosmological view here, whatever. Humans don't matter. Complex life will come back again sometime. No biggie.
What I want to know, this is very important. This can tell us a lot.
Yes. Okay.
Of the species that survived that thermal maximum, how many of them relied on agriculture for their sustenance?
Right.
Not a single one. Not a single one.
Gosh, that's unfortunate.
I don't think there was any agriculture 56
million years ago. We don't have a comparative for our...
nope. There's nothing to compare to now, for sure, really. But...
yeah. So, I mean, it's good news, though. It means that there's capacity for healing. We just have to actually stop the bleeding first. We have to
stop doing the damage.
Yes, exactly.
We have to stop the bleeding. We have to also stop the damage.
Pull out the knife. Yeah.
You don't leave the knife in there and just shove paper towels in around it.
You do it till you get to the ER. But now the scientists are the doctors in this scenario, and they're telling us what to do. We have technology to use. We have other ways to make energy. Let's move over. Let's ditch the carbon, and it'll be tough for a couple hundred years, but we'll get through it. We'll survive. That's what it comes down to. Do we want to survive? We got to cut the carbon.
I'd like to survive, and I'm selfish. I have offspring who I would like to survive as well. So, longevity, perpetuity of the genetic lineage. Yeah, come on, Leverett. Let's do something, people. Come on. Let's make these genes last. All right. I think we've made it to the end of this show. Definitely not to the end of humanity yet. So, yeah. Thank y'all for being here with us. This was fun. Thank you for listening. Yeah. Shout outs. Out. Wait, why am I howling? I don't know. Anyway, shout outs to Fada. Thank you for help with social media, show notes, and social media and show notes. Very, very helpful. Show descriptions and things. Gord, Arnlore, everyone over there in Twitch and other places. Thank you for helping to keep the chat rooms happy, nice places to be. Rachel, thank you for your assistance and editing. And Identity 4, thank you for recording the show. I would also like to thank our Patreon sponsors. Thank you to Richard Badge, Kent Northcote, Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tazi, Woody MS, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Reagan, Don Mundus, Stephen Alberon, Daryl Myshak, Stu Pollack, Andrew Swanson, Fredis104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Sean and Lena Lamb, John McKee, Greg Riley, Mark Hesson, Flo, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard Brendan Brendan Minish, Johnny Gridley, Rummy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Bob Coburn, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Phillips, Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim DePose, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, Ulysses Adkins, and Jason Roberts. Thank you for all of your support on Patreon. And if you would like to become a Patreon sponsor, head over to twis.org and click on that Patreon link. On next week's show,
we will be back 8
I
p.m. Oh, yeah, not all of us.
Most of us will be back.
But some of us will be returning to continue the show Wednesday, 8 p.m. Pacific time, broadcasting live from our YouTube, Facebook channels, and 4 a.m. Central European time, if that's where you happen to be. On Thursday, though, not on Wednesday. The world is weird, plate tectonics time zones.
Hey, do you want to listen to us as a podcast? Maybe while you study the calls of howler monkeys? I suggest you Google them. They're very cool. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today and notes to stories, links to stories, you will be available on our website, www.twis.org. And you can also sign up for a newsletter. I think it might still be. Are we still? Is that? We're maintaining. Yeah, there's still a newsletter. It's not too late. You can still sign up.
Not too late.
It'll come at some time. It's like waiting for Godot. You just gotta, hang out long enough.
Wait.
Maybe it'll come. Maybe it won't. You can also contact us directly. Email Kirsten at thisweekinscience.com. Justman at twisminion@gmail.com or blame at blairbaz@twis.org. Just be sure to put TWIS TWIS in the subject line, or your email will be spam filtered into the tangled quantum hairs of a black hole, and no amount of conditioner will ever get them out.
Never, ever.
No. You can also hit us up on the Twitter where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview haiku that comes due in the night, please let us know.
We'll be back here next week, a couple of us, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. I've got one disclaimer, and it shouldn't be news. That what I say may not represent your views, but I've done the calculations.
Yeah, done those calculations, and I've got a plan. Woo-hoo-hoo-hoo, we're in the after show. Don't you?
A very short after show today.
Yeah, short after show today, everyone. Blair is not feeling well, and I am conferencing. Once a year, I run a conference called Science Talk, which is for science communicators, and it is this week.
Woo!
We have, yes, today was setting up the in-person location.
In other times, I might have found myself in Portland this week, but not now.
Not now. And I'm still all right. So we've been doing lots of virtual workshops and events for the last couple of weeks because we know people aren't ready to come to do traveling and in-person things. We also know some people maybe don't have the money to do it, but we want to give that option to people. But there are a lot of people who do want to get back together and do things in person. And so we're doing that. And today it was very nice because we have this big giant room and there were maybe 5 people in it.
Safe,
And we were
very
working
safe.
we're like across the room from each other, yelling at each other, but there's air. Get the window open. There's a lot of air in the room, and we're setting up wires and cables for the internet, and we've got gig download speeds. I'm seriously, I'm just gonna. Do you guys want movies? I'm gonna download some movies. No, I'm kidding. If I need to download things, is there a link with more details? Yes. If you go to, I will get the link.
Sciencetalk.org. I can put it
Yeah,
in
you
there.
can
keep
go to
talking.
sciencetalk.org for sure. And right up in the top there is a link. Thanks, Blair.
Yeah.
For the SciTalk 22, the conference. You can see past conferences that we've done. But it's really, people who communicate science, trying to figure out how to talk about science better, learn from each other about things that we've done, how we've done it, all that kind of stuff. Community. It's community. No, I'm not talking. Yeah, but it's really wonderful after many months of work to finally see something like this happening. It's a lot of work because it's hybrid, but it's really fun because we keep having Ghostbusters jokes because the AV team I'm working with, they've got the outgoing stream, and we also have virtual attendees, so we have the incoming stream. Sorry, Justin, I didn't see you there. Come back. And so there's the outgoing stream and the incoming stream, and
you can't cross them. Don't cross them.
That's the mantra. Don't cross the streams. So we can't have a moderator in person on stage being streamed out, and then in the same session having somebody being streamed in. So we've had to split them up into these separate sessions because we can't cross the streams.
Can't cross the streams.
It's can't. So every time I've gotten confused. Every time, but it's great. Every time I get confused, I go, "How is this working?" Oh, don't cross the streams, and then I can figure it out.
But if there's ever like a giant marshmallow creature, then that's the thing you would want to do.
Right.
So it's kind of iffy advice. That whole, it's how is the thing
situational, right?
Also, the solution, yeah.
It is situational. Oh, I don't think my picture uploaded from today. I took a picture of. I bought a mascot at the store today because our mascot this year is the Science Talktopus. Yeah, Fada's going to be at the conference.
Nice.
Yes. Yeah. There was a meetup earlier tonight. I did not go to it because I was working on the streams. Setting up the streams. We have Vimeo, we have Zoom, we have Hoova, we got all these streams. Who's in charge of the streams? I am. Where'd my picture go? So I took a picture and of my Science Talktopus. I put him on the stage. I have to get this. Oh, I know what I have to do to get it so I can show you. But I'll have more pictures later after the conference. I'm not going to update Google Photos. How about... There you go. Okay, it's uploading. I can share my little octopus squishy guy. We're going to sit him in the front row to make all of our guests comfortable. He's our emotional support octopus. So presenters won't be scared. Emotional support. Yeah. Yeah, it's funny. I don't really talk about it a lot on TWIS because this is the podcast. I just want to talk about science with y'all.
This is the after show. Welcome to the after show, everybody. We can talk about anything.
We can talk about anything in the after show. Thank you for liking my socks, Garam.
Grumpy B, imagine in the chat room, imagine every molecule in your body simultaneously accelerating from every other molecule in your body at the speed of light. Ow!
No, I don't like that. I'm glad I have my glue-ons. Stephen Rain is studying howler monkey calls.
Good. They're very good. For something called a howler monkey, you'd expect it to sound different.
Okay, I think this means that I must play one for you.
It sounds like this.
Okay,
What?
now we will compare. Okay, we're gonna Blair. And
It's like a mixture of that and there you go.
Okay. Okay. This is from the Houston Zoo. Houston Zoo Television. Hear a howler monkey howl. Come on. Get to it.
Get to it.
Come on. This is very slow.
These are our black and gold howler monkeys. The black ones are male. This is Mowgli. He's
Stop
19
talking.
years old. These are his daughters, Vita and Garcia. These guys peanuts today. These guys, they prefer new leaves.
I just wanna
Species
hear
of
them.
howlers.
Okay.
The cage white rarely do that. These will eat eggs in the wild. We were kind of comforted by that because these guys love egg day. Every morning they do this, and this is because leaves don't give you a lot of energy, so they don't like to expend a lot of energy, so they don't want to have to fight over their trees. So first thing in the morning they do this call. It's usually done around 4 a.m. or just before dawn, and the call allows all the other howlers in the area to sound actually more like a Gregorian chant. These guys, because they have...
I want her to stop
greens, and
talking.
so
I like all the information, but not right now. I just want to hear the howler monkey.
Just want to hear it.
Wow. Oh, I've never heard that. That was great. Wait, where did you go? More howler monkeys. So howler monkey may or may not sound like your neighbor.
Grover Sharma. Okay, that sounds like a horror movie monster. Yeah, they should call it the horror monkeys.
It kind of sounds like a delayed or like an extended burp also.
I think a belcher monkey? This sounds like it's already a thing. Is that already a thing?
Who's to say?
So there's so many creatures on the planet. Strange names.
Mouse pad.
So what's some of the stuff that's coming up at this science talk?
At the science
What are some of the subjects that are going to be tackled?
What
Is
do
it
we have
speakers?
tomorrow? Let me tell you about our agenda. Yes.
What's going on?
What is my agenda? Well, we've got some workshops. People will get a workshop on disaster recovery communications. The workshop is called "We're the Government and We're Here to Help." So it's government communicators talking about recovery after disaster, after the fires and earthquakes and stuff recently. Another person is doing learning engineering, so designing SciComm using the science of learning, basically how we the science of using the science of how we learn to develop better ways to communicate science. And then there's a workshop which is going to be awesome, which they have this group, they have a podcast and you can listen to it, and it's role-playing games, which we've talked about this, but dugongs and sea dragons is what they're called, and they do ocean science but as like role-playing, so they all have characters.
Okay.
And tomorrow they're going to be doing their podcast and their characters and getting the audience to help with like their roles and the things that they decide to do and all that kind of stuff, which I think that one's going to be a lot of fun. We have Senator Lou Frederick is going to give some statements. He's an Oregon senator who I didn't realize actually is a science communicator, which is very exciting. Yeah. Cheryl Kirschenbaum, she wrote The Science of Kissing, is a book that she wrote, but she's also written a book on science politics. She's written books with Chris Mooney. Currently she works on like food and agriculture stuff and does videos for PBS studios. But she started Science Debate is one of the things that she's done. So she's been involved in all sorts of interesting aspects of science communication. We've got a talk on science in a pandemic from the Peruvian perspective, religion and inclusive science engagement, the art of narrative storytelling and its role in science communication. Yeah, there's a whole bunch of different things. And then we're going to have posters, and there's a movie screening and all sorts of fun stuff.
Cool.
Yeah. Yeah, a lot of neat things. I hope that people learn things and gain community, find other communicators to do things with and hang out with. I don't know. Sometimes science communicators, we're all over the place doing different things. We're like, where's my community at?
Yeah, I'm hoping that in 2023, perhaps we can, TWIS can make a comeback at Science Talk. That would be fun.
Yeah.
That would be great.
We'll see. We'll see where Justin is in the world at that time.
Oh yeah, I'm going to something. I'm going to
You're going to something sometime.
Well, no, this weekend I have a plan to go to a Neanderthal talk.
Cool. That'll be awesome.
I think the University of Copenhagen. Which is a really exciting thing. I'm very super excited about it. But it's also going to be like the first time I've gone to a place with humans, group of humans in a long time. So
yeah, that's this for me is this event, and I'm just kind of like
And I honestly think it's going to be too soon, but I feel like I'm always going to think it's too soon. Like at some point, I think I wrote off social interaction at some point.
Yeah, it's very interesting for people that that had to stay out in the world versus people who are able to cloister a little bit. How different it feels, right? So, like, for people who've been going to work throughout the pandemic, there's no fence post. Everything's just always been normal. Throughout the pandemic, just like, oh, I'm allowed to go to the store now? Great, I'm gonna go. So it's definitely, I get it. Like when I was fully shut down and teleworking, going to a store gave me anxiety. But then once I was going to work full time, it was like everything went back to normal in my brain so fast. Took like less than a week.
Yeah, I mean, we are meant to be social creatures, even those introverts among us. We are, going out to the store, going and doing things, having a lifetime of training in those things is not going to disappear in the couple of years of the pandemic.
There's a hump getting it back, but
for sure.
Once it's back for sure. It was an interesting phenomenon for me.
Yeah. I hope to be able to reflect on it later.
Yes.
Like right now, I'm like, oh, tomorrow is going to be crazy.
Yeah. It'll be fine. It's all going to be good. You're in a room full of scientists, so they're all vaccinated, I'm sure. So
they're all, everyone has to be vaccinated. They have to sign a waiver saying they're vaccinated and they don't have any symptoms. And yeah. So fingers crossed that the smart people do the smart things and don't let their emotions, which are going, I want to see the people and I don't want to have FOMO, that they don't let their emotions get the better of them, but they use logic to dictate their actions in the next couple of days. That's what I'm hoping.
And my logic tells me I have to go to bed.
Okay. Yes. And I must as well. Like we said, not a long post show tonight. This morning.
at this moment in time. Say goodnight, Justin.
Good night, Justin.
Well,
say goodnight, Blair.
Good night, Blair. Good night, Kiki. There's a delay.
Good night, Kiki. There's
Is there
a big one.
one?
It was for a second. Or was I just cognitively unaware?
Sometimes I wonder if it is a delay. I don't know. Good night, everyone. Thank you for joining us for another episode of This Week in Science. We are so glad to have spent this time with you. Have a wonderful week. Blair and Justin will be back next week. I'm going to be in Rochester, New York. So if you are in Rochester, New York, check out St. John Fisher's College because I'll be giving a lecture next week. Woohoo! All about the hopeful nature of science. Gonna talk about hope, baby. Gonna talk about hope for you and me. Okay, everyone, stay safe, stay sane, stay curious. We'll see you next week. Bye. Hanging up.